All right, welcome and thank you one more time for joining us today. My name is Carlos Bosques. I'm the CEO at FOSSI. And at FOSSI, our focus is to understand, translate, and intervene with food allergies. And we've established a unique program in a unique collaborative network of leading scientists focused on bringing us one step closer to a cure. In a short period of time, FASI has changed the landscape of food allergy research, and we're excited for the road ahead. This year, we've put together an exciting lineup of speakers for our seminar series, Our Journey to a Cure. Throughout the year, we have been discussing our journey through FASI's endeavor to create a world without food allergies. Please continue to join us for a monthly seminar series where we'll spotlight critical science on the path to developing transformative therapy. Today, we're thrilled to be joined by Dr. Ramnik Xavier, who will be talking about his work studying single-cell gut architecture for the atopic march and associated diseases. Ramnik wears many hats at the Broad Institute, including the Klarman Cell Observatory Director of the Klarman Cell Observatory, Director of Immunology Program and Infectious Diseases, on also the Microbiome Program. Ramnik is also a co-director of the Center of Microbiome Informatics and Therapeutics at MIT, professor at Harvard Medical School, and professor of the Department of Molecular Biology at MGH. So before we start, I just want to remind the audience that we will take questions from our Q&A box at the end of the seminar. So please send your questions and we'll be happy to address them. Ramnik, very excited to have you here. Thank you for taking time out of your very, very busy schedule. It is really an honor to have the opportunity to talk to you today.
Thank you, Carlos, for the invitation. I'm delighted to be here and happy to share with some of our studies that are ongoing to better understand allergy more broadly and how it impacts food allergy more specifically.
Thank you, Ramnik. I think, Ramnik, before we get into the science, always people ask, we want to learn about the journey of the amazing investigators. Would it be possible to tell us a little bit more about your scientific trajectory? Why are you studying specifically this area of investigation, and why food allergies?
Thank you, Carlos. I'm a physician scientist at Mass General Hospital. After completing my residency, I studied gastroenterology, and that got me thinking about the many diseases that affect the gastrointestinal system, where the causative factors are unknown. And the way I've gone about trying to get closer to causality is to ask, are there genes within an individual that increase the risk of disease? Are there modifiers that increase the risk of disease? And obviously, specifically in food allergy, a disease that starts in the gut. I've been interested in trying to better understand the tissue site in which the disease starts. And I will try to illustrate this using a few different examples of this concept called the allergic march. So patients with food allergy could be their first presentation, or they could have allergic diseases that affect, say, for example, the skin in the form of atopic dermatitis, or the lung in the form of asthma, or in the esophagus in the form of eosinophilic esophagitis, so a disease that's in close proximity to the small bowel. And learning from each one of these as starting points is going to advance specific understanding of food allergy, and more broadly, the global understanding of food allergy. And so you might ask, why now? For all of the diseases that I have listed, the number of patients with new diagnosis is increasing exponentially. And this is not limited only to the developed world. It is also happening in the developing world. And so it's time to sort of begin to understand what are the causal factors so that more broadly, the Food Allergy Science Initiative can also start thinking about prevention and treatment.
Thank you, Ramnik. No, it is exciting. You touched a little bit on this, but sometimes we, as FOSSI and all the investigators look at food allergies, we look at it as just food allergies. But one of the things that I'm really excited about the work you've been doing is really looking in the context of the associated diseases and the atopic march, and more importantly, at the site of action. So I think it will be good for people if you can go a little bit deeper into the atopic march, the associated diseases, why to look at it in a chronological order of events and their connections. If you can touch a little bit more on that, would be great.
Yeah, so I think to illustrate some of these concepts, I think I'm going to use a few slides, because I think then people can appreciate what I'm referring to. So if everyone can see my slides. So as I said, food allergy is the big question. Why is the incident, why are the numbers of food allergy increasing in children as well as in adults? Is it your genes? Is it the food you consume? Or is it other environmental factors? As I mentioned, this concept called the allergic march that's been known for many years can start with food allergy and then extend to an organ nearby, the esophagus, and this condition is called eosinophilic esophagitis. And when it impacts the skin, it's allergies of the skin, broadly defined as atopic dermatitis. And then, as I alluded to, it could affect the lung or the nasal mucosa. And these conditions can precede food allergy, can follow food allergy, could happen at the same time. And so broadly, what I'm going to tell you today are about two of these important components of the allergic disease progression. And what I'm going to highlight is everything that I do in my lab or think scientifically always starts with my patient, Dr. Hecht. So I start by learning about the disease from the patients I see. And then by engaging patients and learning from how each individual's disease progresses or doesn't progress or resolves, I begin to come out with questions to try to understand. So a concept that I'd like to highlight for many of these diseases, so often conditions like food allergy, eosinophilic esophagitis, atopic dermatitis, are studied by collecting blood samples from patients. And despite doing these types of studies for many years, understanding of the pathogenesis, of how the disease is initiated, is poorly understood. And what I'd like to highlight today is that the best way to study the initial events of the disease, why the disease progresses in some individuals and doesn't progress in others, why does it become chronic in some, and why does it resolve in others, is to study patients and study biospecimens from the site of disease activity or disease presentation. So the way I've gone about it is to study healthy individuals, study at-risk patients. They're often defined by their genetic makeup or their family history. Those individuals who have early disease, those individuals who have established disease, and those individuals who have resolved disease. So you can see the spectrum that will begin to better understand how do you prevent the disease and how do you treat the disease. Questions that I keep asking on a daily basis are, what are the allergic triggers or what are the allergens? What is the underlying genetics? Why am I born with this risk variant, getting the disease when I'm 2 years old, and in another individual who's not getting the disease? And then are there environmental factors? And as I said, food is the poster child of this initiative. And so are there food factors And whenever there is food implicated in a disease, one often asks, is the microbiome equally important or secondarily important? So that's how I've gone about studying the disease. And just to illustrate this, I'm going to give you one example, and I'm going to start with allergies of the esophagus. So eosinophilic esophagitis is a chronic inflammatory disease of the esophagus. It affects the young, and over the many years that I've been practicing gastroenterology, the number of patients who present with this disease is significantly increased. I've listed below the classical foods that have been implicated in eosinophilic esophagitis. And the diagnosis is made by taking a small biopsy from the esophagus and looking under the microscope. So here again, highlighting the fact that examining blood in an individual to make a diagnosis of eosinophilic esophagitis has not worked so far. But by getting the biopsy from the esophagus and counting the number of eosinophils, one can make the diagnosis. And what I'm showing you in the same slide is a slightly more complicated definition of the disease. So there is a food antigen that disrupts the barrier. This impaired barrier sets up the immune response. The immune system, both the two types of immune system, the innate immune system and the adaptive immune system, is activated to bring about progression of disease. Now, avoidance of these food that I've listed below has been shown to calm this inflammatory response. So that's the direct connection of immunology, barrier function, and food. And if you take children with the disease, or adults, you can begin to see, in a normal esophagus, you have a very smooth lining. And when you get the disease initiated, you can begin to appreciate these red cells coming into the esophagus and initiating inflammation. If you don't take the allergen or the trigger away, then you end up with chronic disease. And that's chronic eosinophilic esophagitis or fibrosis. And then if you leave this trigger to continue, then you end up with a scarred esophagus that's much more narrower, and patients start complaining of difficulty swallowing, chest pain or chest discomfort because of obstruction. And so the goal here is, can you understand the initial events of the disease so that we can diagnose the disease earlier, find treatments that could prevent the disease or maintain the disease, and then avoid this complication that's called narrowing of the esophagus. So what we did was to go and take a small microscopic 2-millimeter biopsy from the esophagus, dissociate all these cell types, and ask what triggers the disease. So what we did was we examined 22 individuals, some with active disease, some with disease who had a diagnosis of eosinophilic esophagitis, who had disease resolved, and then a group of individuals who are healthy. What was striking was, with that small biopsy, we could find many different cell types. So 60 different cell types, and some cell types that were not previously known to exist in the esophagus. Now, I told you that eosinophilic esophagitis is a disease that accumulates eosinophils in the esophagus. What we identified was that at the same time as eosinophils accumulate, now you get a second cell type called mast cells also accumulate. So suggesting that there may be two cell types that could trigger the disease. The second is we also then asked the question, what are the other cell types? And without going into details, you can clearly see that, when you have active disease, you see a very striking increase in the abundance of a specific cell type. And then you can map it to the various cytokines. The cytokines are products of cells that lead to tissue damage or tissue injury. So one potential way to think about treatment is to come up with a molecule that mops up this cytokine so that you can now decrease this inflammation. Now, I told you that while the disease started with eosinophils that the pathologist found under the microscope, but by more advanced methods, we've now been able to identify many more cell types that contribute to the disease. So I think I'm going to stop there and try to answer Carlos's additional questions and then elaborate further on what I just presented.
This is fascinating, Ramnik. One of the things I always like about this is, people have been studying food allergies and different associated indications, either in mice or in the periphery for many years, right, in blood. But like you said, understand directly in disease-relevant tissues, it seems critical. Specifically, and one of the things that resonated as you were showing this is you're getting the eosinophils, but you're getting mast cells, and there it seems like mast cells in the periphery are different than the mast cells that you have in the gut. So it is really exciting to see this. I don't know if you can provide a little bit more detail in terms of the characterization of the disease of not only EOE, but I guess other diseases associated with food allergies in disease-relevant tissues that you're doing in your laboratory. I know as you're stepping into also more in the areas of food allergies and some of that data.
Yeah, so we've done a similar study in children who are at risk of food allergy and children who have established food allergy, where by getting permission, we've been able to go and take small biopsies from the small intestine with the same endoscope. We've also done a similar study in patients with celiac disease. So in celiac disease, you know what the trigger is. So this is a disease that is initiated by being allergic to a product of gluten. And we're building similar maps to what I showed you about in eosinophilic esophagitis, so that we can now figure out, is there a precursor change in the lining of children who are at risk of food allergy? Is their gut lining more leaky than in a kid who is not at risk of food allergy? So by doing these biopsies and what we call single cell analysis, we can come up with those findings. And then similarly, we want to figure out, in a child who has established food allergy, how does this change? What are the specific cell types that change? A child who's initially allergic to peanuts, when he or she becomes allergic to a second and third agent, are there different changes in the lining? And then obviously, most importantly, how can we reverse this process? And could we now learn from children who get out of their allergic state as they age? So that's a learning of nature's experiment. But more importantly, in kids who get OIT as a potential therapeutic strategy, could we now figure out what does OIT do to that lining of the gut? What aspects does it improve? What aspects doesn't it not improve? And as Carlos had in one of his earlier slides, the broader mission of Food Allergy Science Initiative is to identify these trigger points and come up with therapeutic options or preventive strategies. And one additional comment about the FASI is that it's extremely rare to bring physicians, scientists, thought leaders who are actively involved in caring for patients with food allergy, with scientists who are interested in deep mechanistic understanding, to be continuously connected, so that you can now begin to, ask questions from one another and, refine your thinking. Because, as I said, everything I do in the lab with in the scientific side of my career is always starting with patients. I want to figure out what's wrong in patient A versus patient B, and then from that map, begin to think about how the disease is on, off, and doesn't progress.
Thank you, Raman. No, I appreciate it. Sometimes I know it's challenging to study the tissues and patients directly, especially when you're going to disease relevant tissues. But to your point, there's nothing better. If we want to try to find a cure, there's nothing better than going to the relevant tissues and being able to prove some mechanisms in the animal models, but then come back to the humans as well. So really exciting, great data. And we're really looking forward to also a lot of the food allergy data. Maybe before we go, I know there's a few questions. Before I go into the questions and answer session, I want to just remind everyone, we have the Q&A box. Please submit your questions. And want to remind everyone always asks who can they get involved with FOSSI. So donate, host an event, follow us on social media, and join our webinars. We, as we've taken you through the year, a lot of great discussions. Our next webinar would be in the upcoming month on May 7th. And we'll be talking, as we've been talking, about the progression of food allergies and all the mechanisms. And now, as you've heard from Dr. Ramnik Xavier, really studying the disease in disease-relevant tissues in humans and try to identify markers. The next discussion would be about understanding our new initiative for building the drug discovery accelerator at FOSSI and how we start catalyzing the cure for food allergies. Questions. I have a few questions here. I think the first question, Ramnik, is, it's a big endeavor of finding disease-modifying therapies. There's now 2 approved after Xolair was just approved a few weeks ago and Palforzia prior to that. What's next in order to change the landscape of food allergies, especially how do we need to understand food allergies in order to create disease-modifying therapies?
Yeah, so if I step back, I mean, there are lots of attempts on goal, but I think one of the commitments from FOSSI is to also try to reduce therapeutic discovery and the failure rate. So if you look at drug discovery programs, 90% or so of the drug discovery programs fail as you get to phase 3. So an important commitment at FOSSI is start with human biology, learn from patients. And so, as I showed you in eosinophilic esophagitis and with some of the studies that are ongoing with food allergic children, is that we are identifying many different cell types. The cell types tell us about genes that are different between disease versus no disease. And by studying, the biology of those genes, applying tools such as machine learning to those findings, we now can identify a gene list or a target list or a pathway list for which we can come up with, better intervention strategies, whether it be an antibody, whether it be a small molecule, whether it be a peptide. So there are many chemical interventions that one can go in. And so the idea is that, the types of studies I presented is going to, we as a group of FOSSI investigators will pore over the data over and over again so that we can then prioritize, 1, 2, or 3 starting points And since these 1, 2, or 3 starting points have come from patients with and without the disease, I'm confident that not only can we get to efficacy and safety, but also shorten the time from starting a drug discovery program to pill in a bottle, as one of my colleagues used to tell me this often.
Thank you, Ramnik. No, really, it's a long journey, but hopefully we'll get there. I have another question here is, the biopsies, great data, multiple biopsies taken. Are the multiple biopsies taken from different parts of the esophagus, or is it one part? And how do you make the connection to different?
Yeah, so with the endoscope, we can look with the eye and the camera that's attached to it and say that this is normal esophagus and this is abnormal esophagus. Similarly, in the skin, we can look at it and say this is normal skin and this is abnormal skin. And it might be useful if I just show you one slide to highlight this. So what we would do is to take, in this case, a biopsy from a healthy donor, patient with atopic dermatitis. But more importantly, we would take a biopsy from the clean area to show that the non-inflamed part. And then we'll take a biopsy from the inflamed part to basically say this is how the disease trajectory changes. So often sort of 2 small puncture biopsies, less than 3 millimeters in size, is good enough. Now, obviously, when you do these types of studies, you can also find unexpected findings. And if that's the case, then we would take more than 1, 2 biopsies.
And I guess in this case, Ramnik, you almost have, I guess, a control within the study, because you can take areas that are outside of the lesion as well, which is a different type of control because it's still
so you have
Yeah, no, and sometimes that is difficult when doing this.
Yeah, I mean, in another study, in patients with Crohn's disease, a few years ago, we had done a study where we took from the same patient a healthy piece of tissue, an inflamed piece of tissue, and then also a non-inflamed piece of tissue. So we took 3 biopsies there. So we can see what health looks like, what inflammation looks like, and what resolution looks like. So that in the same patient, you capture a trajectory.
Beautiful work, Ramnik. I know we're out of time, but we really appreciate you're doing amazing work, and we're really excited to have you as part of as a FASI investigator, and we're looking forward to being able, to your point, learn from diseased patients so that we can try to generate therapies for patients that are suffering with food allergies. So thank you for your time, Ramnik, and thanks everyone for joining us today in this great talk.
Thanks again, Carlos and FASI, for the amazing support and bringing together an amazing community of investigators, all of who are committed to sort of getting to causal factors and hopefully treatment strategies to help every patient.
Thank you. Thank you, Ramnik.
Okay, bye-bye.
Thank you, everyone.